Hello and welcome to this video on section 14.6 on DNA repair. Our learning objectives here are to explain DNA repair mechanisms and discuss the different types of mutations in DNA. So first we have to note that DNA polymerase, as it is replicating our DNA, has a proofreading ability and it rarely makes mistakes, so it's pretty efficient. It is going to read the newly added base before adding the next one, so that if it does make a mistake, it can go back and put in the correct one. And to do this, we're going to utilize a 3' exonuclease so that we can cut that phosphodiester bond and release the wrong nucleotide and replace it with the correct one. So what happens when we have a mismatch repair? If we don't detect that error during replication with our DNA polymerase, we can correct it after through this repair process. We are going to find our mistake first. So this is our mistake over here, and excise it and then resynthesize it. We don't really know how it does this, but we know that it can be repaired. And if it isn't, then permanent damage can be a result. The other type of repair we have is a nucleotide excision repair, and this is when we attempt to remove the damaged bases. So for this case, we're going to utilize an enzyme known as a nuclease, where we are going to cut both at the 3' and the 5' end and essentially get rid of that damaged base. That DNA segment is going to be removed, and then we can bring in DNA polymerase so we can add the correct nucleotides. And then our gap is going to be sealed using our glue enzyme, which is our DNA ligase. So it helps to bring those phosphodiester linkages back into place. An example where these mistakes are not corrected are in people that have xeroderma pigmentosa. And this is a skin condition in which we are highly sensitive to UV rays from the sun. And unfortunately, people who have this have a higher risk of skin cancer. And what's going to happen physiologically is that when we are exposed to those UV rays, they are going to have our pyrimidine dimers, especially our thiamine, form, and this destroys the structure of DNA. It kind of reminds me how we saw this little bubbling happening here that we do not want in DNA, and unfortunately we can't repair the damage. And we may also have a defect within excision repair enzymes that didn't allow it to be repaired in the first place, which can then lead to damaging of the skin. Now, the last thing we're going to talk about are mutations, and there are so many different ones that we can discuss. So we just have the most common ones listed here. And when we say mutation, we're talking about that there's some type of variation within our nucleotide sequence of our genome, and usually it's due to some type of DNA damage. So that damage can be either induced or spontaneous. Induced means that we've got some type of exposure to a chemical, UV rays, X-rays, or other environmental agents, whereas spontaneous happens without any of that exposure, and it could be a result of natural reactions just taking place within our body. So let's first talk about point mutations. There are two main types. We've got our transition substitution, where we are going to have our purine or pyrimidine replaced by a base of the same kind. So for example, adenine is going to be replaced by guanine. Whereas our other type, transversion substitution, is where our purine is going to be replaced by a pyrimidine, or vice versa. So for instance, a cytosine is replaced by an adenine. And sometimes we can see that these mutations have no effect. So here we see our original sequence. And then we see that, let's see, where are we at? The adenine here was replaced by a thymine. So what would that be an example of? Would it be transition or transversion? Well, in this case, adenine is our purine and it was replaced by a pyrimidine. So this would be an example of transversion. If we have a missense, then that means we are going to have a nucleotide put in place of another one. So in this case, the cytosine is replaced by an adenine. Do you remember what type that would be, transition or transversion? If you said transversion again, you are correct. And then here is a nonsense where essentially a stop codon gets produced. So here we see tyrosine, and that last letter, the third letter, was changed to a G. And this change caused a stop codon to be coded. And we went from a pyrimidine to a purine, so this also would be a transversion. And this means that it basically will terminate the protein early, so it may be non-functional at this point. Another type is called a trinucleotide repeat expansion, where we get an increase in the number of copies of the same codon, and we have repeated regions within the same amino acid. We also have insertions or deletions. So this is exactly what it sounds like, where we are either inserting nucleotides or getting rid of some nucleotides. And so as we are reading that, it shifts the frame. It's like what we saw earlier in the previous sections, where the nucleotides or codons that are unaffected are going to stay the same. But as soon as we start inserting, I'm trying to double check. Yes, this is an insertion of two nucleotides that shifts the frame, and we are now coding for possibly different amino acids. Notice how the insertion of these two amino acids did not change valine, but it made everything else shift down, which then changed the other two amino acids in this mRNA strand. Also, I don't have a picture of it here, but sometimes we do have a piece of DNA from one chromosome get translocated to another chromosome. It could either be to a different region or the same region, and that is known as a translocation. And that is pretty much it for this chapter. Let me know if you have any questions.